Greetings to one and all watching this. My name is ARINDAM CHATTERJEE, an MSc Microbiology student from Central University of Tamil Nadu. Today I will be presenting the topic of marine microbial community with special emphasis on phytoplankton. Let's begin. First of all, what is the marine microbial community? It is the amalgamation of all the microorganisms present within the marine habitat, which can be a sea or a deep ocean. It consists of bacteria, archaea, fungi, viruses, and phytoplankton. Now, what are phytoplanktons to be specific? Phytoplanktons are microscopic, photosynthetic, aquatic microorganisms which provide the basis of the marine food web and produce about half of Earth's oxygen. And they have a very vital role in the marine habitat that they exist in. Now, these phytoplanktons are consisted of both prokaryotic organisms and eukaryotic organisms, but they are all autotrophic or photosynthetic in nature. In prokaryotic organisms, we can see cyanobacteria, which are also called blue-green algae. And in eukaryotic organisms, we can mainly see unicellular algae. Firstly, we have diatoms, which have silica in their cell walls. Then we have dinoflagellates, which have a biflagellate nature. They have one flagella in the transverse manner and one flagella in the longitudinal manner. Then we have euglenoids. Next, coccolithophores, which have calcium carbonate deposits on their outer cell walls. Then we have green algae like chlorophyceae and prasinophyceae. And lastly, we have silicoflagellates. Here is a picture of the different types of phytoplankton colonies that you can observe in a normal marine habitat. You can see some of them are existing in an unicellular state, while some of them are existing in a colonial state. Now, the phytoplankton group consists of different types of microorganisms, and each of those types of microorganisms have their classification system on their own. Now, when we are considering phytoplanktons in general, this group has a classification system on its own. Its classification is based on size, cell wall compositions, the light harvesting system, and the composition of pigments. First, let's begin with size. Now, the phytoplanktons whose diameter ranges from 2 micrometers to below 2 micrometers, they are called phytoplanktons. Next, we have those that have a diameter of 20 to 20 micrometers, called nanoplanktons. Then we have microplanktons, which have a diameter of 20 to 200 micrometers. The size is very important for these microorganisms because it influences their surface area available to the environment. Now, since we know they are the autotrophic or photosynthetic organisms, so we can understand that the size directly reflects the amount of surface area they have available for absorbing sunlight and performing photosynthesis. Also, these organisms not only need sunlight and CO2 for their growth, but they require other nutrients as well. So the size also dictates the surface area which is available to the environment for absorbing these nutrients and growing. Nextly, the size dictates how much surface area is available, which also dictates how much time they can float on the surface or the top layers of the marine habitat, and how fast they will sink into the ocean. As we know, spherical microorganisms, they have a lower surface tension, so they can sink faster, whereas those that have a flat configuration will stay afloat for a longer time and will be able to photosynthesize for a longer duration. Next, we have cell wall composition. As we already said, diatoms, they have silica or pustules in their cell walls. Dinoflagellates contain cellulose plates. Coccolithophores have calcium carbonate deposits on their outer cell wall, and cyanobacteria and green algae have polysaccharide-based cell wall matrix. This is a diagram which shows the different size variations of phytoplankton groups. Next, we have the composition of the photosynthetic pigments. Now, all these photosynthetic organisms have one pigment in common, that is chlorophyll A. The difference arises when it comes to the accessory pigments. These accessory pigments are responsible for absorbing other lights of different wavelengths and making them Next, we are coming to photosynthetic pigments. Now, all these organisms have one pigment in common that they have for light harvesting. Now let's come to the photosynthetic pigments. Now all these photosynthetic organisms have one pigment is common, which is a primary light harvester. The difference arises when it comes to the accessory pigments. Firstly, we have fucoxanthins, which are mostly found in diatoms. Then we have peridinin, found in dinoflagellates, phytoplankton, which are found in cyanobacteria, and chlorophyll B and C in some green and golden algae. These pigments expand the range of light which can be absorbed by the microorganisms and can be then used to reduce CO2 into organic forms of carbon. Next, we have photosynthetic apparatus. Now, we have already seen that these phytoplanktonic groups are consisting of prokaryotic and eukaryotic organisms. Now, prokaryotic organisms have a photoharvesting system that is present on their cell membrane, whereas the eukaryotic organisms have a photoharvesting system that is present in specialized organelles inside the specialized organelles called chloroplasts, specifically in their thylakoid membranes. Next, let's talk about the distribution and abundance of these organisms. Firstly, we will talk about spatial distribution, meaning where in the map they are present more and where they are present less. So as we can see, coastal waters have a higher concentration of phytoplanktonic growth because these areas have rivers dumping into the ocean, and the rivers carries a variety of nutrients with them. Then we have the open ocean. The open ocean is very far from the coastal areas and from the river runoffs. As such, they have very low nutrient composition. And as we have already seen, that these organisms not only require sunlight and CO2 for their growth, but they require other nutrients as well. As such, these open ocean areas have very low concentration or densities of phytoplankton growth. Into temporal variation or seasonal variation. So the most commonly seen blooms are spring blooms, which occurs due to nutrient mixing occurring at the interface of winters and summers. During the summer season, there is a phenomenon called stratification. What happens during this is, if we consider an ocean, a column of the ocean waters or the ocean habitat, now the upper layer is exposed to the sunlight. When the sunlight constantly falls on this upper layer gets heated up, while the lower layers remain cold enough. So this difference in temperature causes difficulty in the nutrients which are arising from the ocean floor or the sea floor towards the surface and being available to the phytoplankton groups. As such, during the summer season, the stratification occurs more, which leads to a decreased nutrient supply from the base of the marine habitat to the surface organisms. As such, their growth is very low. Then we have autumn blooms. When the stratification begins to lower a little, the temperature begins to be common for the whole oceanic column, and as such, the phytoplankton group starts to grow. Now, this problem is usually seen in areas which have temperature shifts, but tropical regions which have almost a constant temperature throughout the year do not see these fluctuations. Next, we have environmental controls. Now, temperature directly affects the metabolic rates of microorganisms. Now, as we know, different microorganisms have different temperature ranges in which they can grow optimally. As such, the temperature variations in different regions will affect which type of microorganisms which are able to grow as phytoplanktons were. Next, we have light intensity. Since they are dependent on photosynthesis for their primary energy as their primary energy source, the areas which have a higher photosynthesis, higher light intensity will harbor more phytoplankton organisms compared to areas which have lower sunlight available to them, lower light intensities available to them. Next, we have discussed what are phytoplanktons, what are they consisted of, and where they are found. Now let's see why they are of so much importance. Firstly, they are the primary producers of the ocean. As we know, every habitat has these primary producers which forms the base of the local food web, where they provide most of the energy that is required for the growth and survival of the rest of the organisms or the life forms in that area. Now, these phytoplankton groups take up the atmospheric CO2 and converts it into organic form of carbon, which is then made available to the other organisms living in the same habitat. Now, they not only provide the organic form of carbon, but they provide another important thing as well, which is oxygen, both in its dissolved form and in its gaseous form. The dissolved oxygen, which is produced by the result of photosynthesis, is made available to the fishes or other marine organisms, higher life forms, whereas the one in the gaseous form is made available to all life forms on Earth. So they are responsible for nearly 50% of the global oxygen production, sustaining life on Earth. Next, we have the foundation of marine food webs. As we said, the primary producers produce the energy and nutrients required for the growth of the subsequent organisms. In this case, in a marine habitat, we can see that phytoplanktons will be consumed by zooplanktons, and these zooplanktons will be consumed by further fishes, and then fishes to bigger fishes, and all like that. And when these fishes will die, their organic matter will be settled to the ocean floor, where it will be made available to the organisms in the lower strata of the marine colony. Now, interaction with zooplanktons and higher organisms. As we said, these organisms form the basis of food webs. The difference in their species composition will be directly reflected in the biodiversity of the overall marine ecosystem. Next, we have the very important one called carbon fixation and export to deep ocean. Now, we have already seen through photosynthesis, they can convert inorganic carbon dioxide into particulate organic matter or organic form of carbon. Now, when these phytoplanktons die or are consumed, this carbon is transported to the deep ocean floors where they are settled, and due to the high pressures in the lower areas of deep oceans or sea floors, over the years they get converted into fossil fuels. Now, this process called the biological pump will be explained in greater detail in subsequent slides. Now, why this process of biological pump is very important? As we know, we have the problem, currently we are facing the problem of global warming, in which greenhouse gases like CO2 are stopping the sunlight or reflecting back the sunlight towards the Earth's surface, which is making the planet hotter day by day. As such, it is affecting biodiversity and life all over the Earth. So what these organisms do is, they decrease the atmospheric concentration of CO2 by using them up for photosynthesis, and then these are consumed by zooplanktons and subsequently by fishes. When these organisms are dead, they remain stable to the lower sediments of the marine column, and then the bacteria present in the lower layers decompose them and consume them for their growth and survival. The organic matter which is remaining gets settled onto the ocean or sea floors, where they are buried and stored for almost hundreds and thousands of years. Next, we have the role in global elemental cycles. Now, these organisms not only participate in carbon cycles and maintain the carbon levels, but they have their roles in other biogeochemical cycles as well. Firstly, we have nitrogen cycles. We have seen that the phytoplanktonic group is consisting of cyanobacteria, and they are one of the known nitrogen fixers. What they do is they consume the atmospheric form of nitrogen and convert it into a bioavailable form, which can then be consumed by the subsequent life forms present in the lower strata of the marine habitat. Then we have sulfur cycle. Now, these phytoplanktonic organisms which are living in oceans and sea floors, these oceans and seas often have a higher concentration of salt around them. As such, these organisms face a constant osmotic stress. So to combat this osmotic stress, they have a survival mechanism in which they produce certain molecules inside the cell called osmolytes. This balances the intracellular and extracellular solute concentration, and as such, saves the cell from desiccation. So one of these osmolytes is DMSP, or dimethyl sulfonylpropionate. When the organisms producing these molecules die off or they are consumed by other organisms due to enzymatic activity or natural oxidation, they are broken down into dimethyl sulfide, which is a volatile gas, and it rises through the marine strata towards the atmosphere. In the atmosphere, this gas is subsequently converted into a sulfur derivative, where they attract water vapor molecules towards them, and as such, they have a high role in the process of cloud formation. So wherever the concentration of this gas will be higher, there will be higher chances of cloud formation in that area. Next, we have iron cycle. As we know, the photosynthesis is not only performed by a single enzyme, it is performed by a cascade of enzymes, and some of these enzymes require iron in them. So these organisms take the iron available from the surrounding habitat and consume them, and after they have died, this iron is released back into the habitat. Next, we have the environmental factors which influence phytoplankton growth. Firstly, we have light availability. As we have seen, that since they are photosynthetic organisms, the light intensity will determine which type of organisms are able to grow in that environment. But the light is not always beneficial. High intensities of light can often damage the photoharvesting systems of these microorganisms. As such, they are survival mechanisms in which they can travel vertically through the ocean strata to determine or to take up the some amount of sunlight which is just adequate for them. They also have the system to adjust their pigments to optimize which light of which wavelength is being captured and how much energy is being generated from that. Next, we have nutrient limitations. This one also we have discussed that they not only require sunlight and CO2, but other nutrients like nitrogen, phosphorus, iron, and silica as well. So any limitation in their nutrient availability will be affected in their species composition. And when the species composition is affected, the overall biodiversity of the marine habitat is affected. Next, we have temperature and pH. Now, this one we already know that different microorganisms have different temperature optimums in which they can grow and survive optimally. So, any change or fluctuations in the temperature will result in the change in the species composition. You can see warmer waters favor cyanobacterial growth, whereas cooler waters favors diatomic growth. The same with pH. Every organism has an optimum pH range in which they can survive optimally. Or alkaline environment will support different compositions of phytoplanktonic growth. Some pHs will impair the growth of certain microorganisms while promoting the growth of others. Next, we have eutrophication. It is a process where the large number of nutrients is suddenly made available to the sea or ocean due to the agricultural runoff water that is being transported by the river directly to the ocean, or through the industrial waste, which contains large amount of nutrients that are being dumped into the ocean or into the river. This can trigger high dense phytoplankton growth, which can sometimes become toxic and can negatively affect the subsequent higher organisms present in the marine habitat. Next we will see that how climate change is affecting phytoplankton ships and why we should be aware of it. First, we have shifting community structure. We know that due to global warming, the temperature of our planet is increasing year by year in a gradual manner. As such, the water bodies in our planet are also getting warmer day by day. Now, this warming is changing the species composition of the phytoplanktonic groups, and which is directly reflected in the higher species in that same marine habitat, since they are indirectly dependent on that phytoplanktonic community. Next, we have ocean stratification and nutrient limitation. As we have already spoken about stratification, where the top layer of the marine habitat gets warmer, but the subsequent layer remains cooler. And this impairs the movement of nutrients from the base of the habitat toward the surface, which can then be used by the surface organisms to grow and survive. Now, this stratification is increasing day by day due to the planet growing warmer, and as such, it is limiting the transport of nutrients and negatively affecting the phytoplanktonic growth, which is again then reflected into the biodiversity of the total of the marine habitat. Next, we have geographical range shifts. Now, since the open oceans are facing these warmer temperatures, some phytoplanktonic species are seen to be expanding towards the polar regions, where the waters are relatively cooler. This is affecting the open sea or the open ocean marine biodiversity. Next, seasonal bloom time changes. Now, we all know that these marine organisms, not only phytoplanktons, but the higher organisms like fishes, octopuses, and all these organisms have a certain life cycle, have a certain life pattern where they will produce eggs in a certain state, these certain time period. These eggs will form larvae, they will feed on zooplanktonic organisms, and then they will grow into mature fishes or mature organisms. Now, since due to climatic changes, the seasonal timings are changing, this is affecting the growth of the phytoplanktonic groups. The phytoplanktonic groups zooplanktonic composition, which is indirectly affecting these higher organism life cycles. So, to conclude, we will see the summary of what we have learned. We have learned that what are the marine microbial community and what is phytoplanktons in general, what are the organisms they are comprised of. Next, we have read about the structure and functions of different types of phytoplanktons, ranging from their size, their cell wall composition, their pigment composition, and their light harvesting system. Then we have seen the distribution and growth of different types of phytoplanktons around the earth. Then we have studied why they are so important, what is the main role they are playing in the environment. And lastly, we have studied that why the current environmental scenario is affecting the phytoplanktonic growth and why we should be aware of it. These are the references I have used for this presentation. Feel free to go through them at your convenience. Thank you very much for your patience.